situ. I need to define it because the phrase in situ was used in the literature sometimes in a very diverse way. So in situ, I believe the phrase itself originally means in the original place. So in situ diffraction, I would define it as you shooting the crystal in the original place where the crystal grew without breaking the seal of the crystallization chamber. So that is my definition. Once you break the seal of the crystallization chamber, it's no longer in situ anymore. At the most, you can call it a quasi in situ. But this is the device in the general format. It's like this. The hardware already changed. I will show you later. But basically, there is a circular window in the middle. The crystal is actually scattered all around this window. And X-ray will penetrate this window directly and shooting at the crystal. And the key point is this window itself is another crystal. We chose to use the monocrystalline quartz. Monocrystalline means it is literally another piece of a single crystal. So it is not a amorphic or amorphic quartz glass. That's very different from that. It is a single crystal quartz. Okay, both the entrance window and the exiting window, the front and back windows are single crystal quartz. And this material contributes very little diffuse scattering. And these 4 panels I showed here are the comparison between this material, the diffuse scattering between this material versus the other glass capillary. For example, it's here, some sort of plastic, COCR kind of a plastic is shown here. And then on top, this is a perfectly aligned single crystal quartz. And then this one is a misaligned single crystal quartz. And then you can see there will be very strong Bragg diffraction coming out. So those a few Bragg diffractions can be avoided completely. And another advantage of this material is it is a good vapor barrier, as good as regular glass, because the ingredient in it is literally the same thing. And we chose this for room temperature diffraction. And next graph shows you the scattering power of scattering curve. I apologize that my software missing a plugin piece, and then it messed up my micron symbol. These are all should be micron. So there are 2 panels. One is per device, the other is per unit thickness. Different materials behave very differently. For example, the COC plastic, very often in the literature, they call it is a X-ray transparent. What do they mean by X-ray transparent is the per unit thickness. If you normalize the thickness of this material, it would not scatter very large. Compared to glass, it scatter very, very little. But it's not very realistic to make a device out of this material to a very thin device. You will have to make it a couple of hundreds microns, even more. Instead of glass, we know the glass capillary, they can pull the glass to about 20 micron thickness in wall. So, and therefore, it's realistic to make a 20 micron to 30 micron thin device. So per thickness, glass looks really bad, but if you can make a thin device, it's not that bad compared to plastic. Okay, here I want to emphasize on this blue line. This is single crystal quartz. So per thickness, it scatters very, very little. Well, the reason why it scatters very little is because there is no water or any kind of disordered material in this material. It's literally a single crystal. Other than this very ordered material of single crystal, there is no disordered material in it. Okay, there's no solvent in any of these. So it does not scatter X-ray. And also you can avoid the Bragg scattering. And since its quality is just as same as glass, you can make a very thin device out of it. 50 micron is totally realistic. And therefore the total scattering here is also virtually none. Okay. The way to avoid Bragg scattering is this. So I will not get into too much of crystallography here. This material has a 5 angstrom long unicell, very small unicell, and therefore what does it mean is it has a very large reciprocal cell. And then here, those array of dots represents the reciprocal cell. So within our interest, the diffraction limit of a protein crystal, resolution of our protein crystal, there are only a few such potential Bragg reflection that could give us trouble. The strategy is to thread this diffraction condition here represented by the red circle and the blue circle through this array of potential Bragg reflections. And it ended up, it will hit none of them. So it will miss all of them. So no matter which way you look at it, if you miss all of them, then there will be no Bragg reflection occurring, which is to say none of these Bragg reflection will meet its diffraction condition. Of course, within the interest of protein diffraction, resolution of a protein diffraction. Of course, beyond that, it's totally possible that we'll hit Bragg reflection, but who cares? We're not interested in that resolution range. Also, if we want to do oscillation instead of Laue diffraction, it's possible to do that as well. Given a X-ray photon energy and the wavelengths of an X-ray, let's say 1 angstrom here, this calculation shows you that we could oscillate back and forth 6 degrees, plus or minus 6 degrees, before you hit a reflection. That is to say, if you rock this array of reflections back and forth 60 degrees, there are some reflection here may get into the diffraction condition, all right? So we can rock plus or minus 6 degrees before this reflection, 100 at 4.3 angstrom will get into the diffraction condition. Or this one at a 2 angstrom will get into the diffraction condition. But if you say, okay, I'm interested in more than just 2 angstrom, then you will have to choose 11 keV at this point, so that this one, at the blue one at 1.5 angstrom resolution, will get into the diffraction condition before after you rock more than 4 degrees back and forth. So it's a reasonably large oscillation range to do monochromatic oscillation. But of course, compared to Laue diffraction, there are the shortcomings of mono oscillation is obvious in time resolution. So our work is mostly on Laue diffraction at biocars. Another advantage of this material is that you could engineer some kind of a pattern, and then you can wet etch this on the surface of that material. And then this is what I tried before. This device is centimeter size, and this hole is millimeter size. And then this is a computer simulation that an edged hole should look like that. In reality, I did achieve something very similar, except that the bottom of that hole is not flat enough. But we want to avoid all of these. It's just the potential. Okay, we concentrated on whether we can crystallize it on this kind of device. And yes, we can crystallize, we get large crystal, small crystal, micro crystal on it. Your question? Okay, can I continue? There are many, in our lab, there are many kinds of protein crystals already being successfully crystallized on this kind of a device. And I cannot even remember all the names of these, but you find many of them are colored because we are interested in photoreceptors that will carry different kinds of cofactors. And then this red background and blue background is due to the safety light of that particular type of protein. So we have to image them under the safety light. And so if you put this device with protein crystal already grown on it in the beam, so this is a biocar's picture in the early days. What is going to happen is the X-ray beam, the incident X-ray beam, will have to go through the front cover. So when it goes through the front cover, there are a few things will happen. It will absorb a small portion of it, but who cares? That attenuate the beam slightly. But this material would not scatter that X-ray if you align it very well. And it will not diffract the X-ray either. So as I showed before, there's no scattering, there is no Bragg diffraction. Okay, after that, the X-ray beam will penetrate the solution. That's where the diffuse scattering coming from. And then will go through the crystal. That's where the diffraction of our interest come from. And then the exit window is more important than the entrance window. That the exit window needs to be thin so that the absorption or attenuation of the reduction of the signal will be minimized. So that's basically what's going to happen. And here I can show you one of the monochromatic oscillation picture derived from such a device. And as I promised that there's no Bragg diffraction of the quartz. Okay, all the diffractions are from. And
what is the resolution of this picture? Like, what is the resolution of the?
Well, you can look at the water ring, and then if you look at the diffraction is not particularly high, probably 2 angstrom. This particular crystal is about 2 angstrom, I would say. And this is a Laue diffraction from this kind of a crystal called the PHRB. And again, there's no Bragg diffraction of the quartz. And then there is another example of Laue diffraction of 7.5 microsecond. And then this zooming panel is from this corner. So you can see there's still one row of reflections. There's one row of reflections here. It's clear at the very, very corner of that image. Okay, it looks like this kind of device could be useful to scale up Laue diffraction in a very dramatic way. So the question is the automation, that whether we can fully automate this operation. Okay, this is the current design of the platform. In green, we represent the device, and the crystals will be scattered on the surface. And then if you remove the beam stop, we can put in infrared LED behind it. And there is a digital camera looking upwards, and then through a 45-degree prism mirror, and therefore we can look at the crystal and image the crystal along the X-ray beam. Okay, that's the first thing we have to achieve. And then these subsystems, the illumination, the pump laser, the translation stage, and then our imaging system, and then the beamline components, or the subsystems that will be controlled and communicated by this Raspberry Pi microcomputer. This is a very cheap computer, and then communicate with all the users, multiple users that is in this experiment. So again, this is a chip holder. This is different design of the current version of the chip holder, and then we put this whole device on this kind of a carrier, and we mount this chip holder here on a translation stage, two-dimensional, two-axis translation stage. And then the X-ray coming from the collimator behind this window. And then this is the beam stop. And then underneath, this is the objective lens looking upwards through a 45-degree mirror. If we remove this chip, this is a 45-degree prism mirror. We look into the X-ray beam, you can see the pinhole, the final pinhole of biocars where the X-ray beam coming out. So there's a narrow slit that the X-ray beam will going through this mirror. At the same time, we can look at the crystal from underneath. So first thing to do, I hope you can see this movie. The first thing to do is to image the whole device. While this device is, the translation stage is carrying the device into array of positions. At the same time, here, the objective lens, the camera is taking pictures, a few dozens of pictures. Okay, everything could be done in complete dark. Okay, I had to turn on the light when I shoot this video. So what we get is an array of pictures of the device, and they may not be aligned very well. See, it can be inclined and so on. But it's guaranteed that there is a little bit of overlap, both horizontally and vertically. And then by calculating the correlation between the overlap, we can pile them together. Becomes a very high resolution montage. This is about 1 micron per pixel. Depends on the setting on the lens. And then here is one example of such a tiled image. This is a tiled image. You can see there's a drop. There's a lake of crystals and scattered many thousands, I would estimate. And then here, this one tile is here. There's the blue outlined. This is the one tile. And then here's one tile of crystal. And then you can see, you may recognize, oh, this is a good crystal and this is a good crystal. And then the arrow points at those short crystals. These crystals are actually more standing up than the good crystal you can recognize. The reason why they are good is because they change the orientation. Otherwise, there will be all crystals laying on the floor. And those standing up crystals are more precious, and they help to fill the reciprocal space. So then what? After we get this, we developed some computer recognition algorithm, so that these are the raw image. Both of them are the raw image. And then we can recognize those line segments in different colors, means at a different stage of the auto recognition. So I'm not going into too much detail of how this is done. This is an entirely different topic it becomes. And then after you recognize those crystals, then of course you can plan your shots. This is where users can come in. There are many possible strategies the user may or may not like. Depend on, this is directly related to the science they might be interested in. But anyway, the shots planned here, I distinguished them with two different colors. One is pink, that represents a primary shot, which means the very first shot on that individual crystal. And the yellow circles are the subsequent shots. The user may or may not want to take them. If we can plan the shots like that, and then we can choose, say, one of the strategy will be a dark light contrast. Let's say the purple one, we will plan all the dark shots, which means before any laser illumination. And then the yellow shots will be after the laser illumination. That's one possibility. I'm not saying you have to do that, and we are doing that. This is one potential strategy you may want to use. And then this is these are two movies. I hope you are seeing them. They should be playing at the same time. So you can see this is actually during the data collection. So the device are translocating to visit each target that we planned. And at every stop, a X-ray pulse will be fired, and so that the detector can read out a diffraction image. And then we can do this at a really large scale. So with one button start, we can literally collect thousands of images. Okay, I guess you get the point. And then here, I illustrate one of the potential strategy. It's called the traveling salesman route. This calculation finds a near minimum travel throughout each planned crystal once and once only, so that the travel will be minimized. And then we find this is the fastest way to go through all the crystals on the chip. But for some reason, some scientific reason, this may not be the optimal strategy. There are competing strategy will be say I want to rank the crystal from the best quality to the least quality, and then give priority to the best crystal at the beginning. That's another very sound strategy. But unfortunately, they cannot be combined. Well, maybe some users will find out a way to combine them. There are other minor things I want to mention is that look at these crystals are recognized. These are obviously large, good crystals, but often there are crystals that are not very large, and then they are clustered together. And then you may also want to shoot some of them, not all of them. But when things getting even worse, there are areas that's really disaster. There's no way you can get quality data out of it. And this is a trick I learned from the single particle cryo-EM. I just doctor this image and just block it, pretend there's no crystal in it. So the software will not visit any of them. That will save a lot of trouble. That's another potential strategy. But anyway, with this kind of large operation, we can easily get hundreds to thousands of Lloyd diffraction images, very good Lloyd diffraction images. May I also mention that we find, I do not have the hard statistics yet, but my impression is because I have done Lloyd diffraction for decades. The in situ diffraction significantly improves the mosaic spread, reduce the mosaic spread of the crystal, because every crystal will only diffract once at its virgin state. So that diffraction usually is the best at the very first exposure. So people usually use hit rate to evaluate such a serial protocol, which means out of this many images you take, what's the fraction? How many percentage of the image that you take that actually contains diffraction signal, that actually you can use the signal. We argue that there is another way to evaluate. Maybe both matrix needs to be evaluated. That is, out of how many crystals you grow, you prepare. What is the fraction of those, all the crystals that you prepared that actually contribute to a diffraction signal? That is called the yield. And the yield is very often used in agriculture and fishery. And another point I might mention is that since the data set, the size of the data set can contain, I will show you this later, but one data set now can contain hundreds and even thousands of images. The very best diffraction quality and the very worst diffraction quality eventually merged into that crystal can be very, very different. Okay, this can be seen from the image quality visually here, and also can be seen by the scale factors and so on later on. So first question, one of the important question people might ask is there a preparation of orientation? As I mentioned early on for these standing crystals. These are the orientation distributions of one of the data set, more than 800 crystal, which means 800 images from 22 devices. It's not that obvious that this type of crystal has a preference of orientation, which is a good sign. And if you do Laue diffraction, and then we all know that the wavelength normalization, the quality of the wavelength normalization is very important. And then these are the wavelength normalization curves we derived from actual serial Laue data. And these are a small portion of a protein that shows you the active center where the water is interesting in this particular project that shows the water. And then here's the FAD cofactor for another protein. I'm not going to get into science, okay, in this talk. And then here is a comparison between a regular cryo monochromatic oscillation from one of the photoreceptors that we are working on. This is a really difficult case, even for oscillation. The structure is solved, but as you can see, a lot of bulky residues are not really in the electron density. And then we also did the serial Laue at the room temperature. The room temperature structure seems to improve those density residues. And then the refinement looks reasonably good. But I have to emphasize, this is really a very difficult project. I can show you another project that is not published in any of these papers. The refined unicell length between A and C varies quite a bit from the percentage variance. You can see maybe plus and minus 0.5% on both ways. So that's another statistic that show there are potential non-isomorphism between so many crystals. Okay, so if you collect data from many crystals, this is a potential problem you are going to run into. But we do not have a hard evidence to show this non-isomorphism already getting to the way and then make some damages yet. We are very cautious about this. These are the other refined parameter detector parameters. For example, the distance from the crystal to the detector and the pixel size of the detector. It always vary a little bit, but it looks it's in check. And the direct beam distribution, the direct beam position off by a couple of pixels, and the detector tilt angle is plus and minus, I would say, 0.5 degree. So this is, again, it's a orientation distribution. It looks, there might be some preferred orientation. But since you have so many crystals, there's no reciprocal space as a consistently missing chunk. Again, this is the wavelength normalization from several of our Laue runs. And I can explain this gray curve, which shows particularly narrow bandwidth. This is probably because when the Hilo chopper of biocars went out of service, so Rob will have to close down the front end slit a little bit in order to limit the total flux so that we can take a long exposure. And then I suspect this is because we only accepting the central cone of the radiation that causes the particularly narrow bandwidth. But I cannot explain why June of last year, there is a particularly wide bandwidth. But anyway, as I'm showing you, all these potential systematic issues will be completely resolved by by the software. And then here, this is a table you might be interested in. That is 3 dark data sets plus 16 light data sets. And then each data set with how many devices that contribute to that data set. Some of them only 1 device. So you put on 1 device, you scan 1 button, you're done with it. And then some of them will be 2 or 3, and other runs will take 9 of them there because Crystal simply didn't grow very well. And we collected this many images from all these devices. And then these are the images eventually merged into the final data set. And this rate, I would call it the index rate, right? So this is not only the hit rate, but the index rate out of how many images you actually really used, how many images. And then overall, it's about half. And most importantly, you may be interested, well, are they good? Are they good enough to give you signal? So all I show you here is this is signal. These are the noise. And then the ratio are the signal to noise ratio. And then the signal to noise ratio. And without getting into the science, it will take too much of time to talk about that. Acknowledgement. All the work are done in Xiaojing's lab, and the biocars are the main facility that we used. We also used a little bit of 21IDD, the life science CAT, and Scott Wayne did a lot of work on the automation and the data analysis. And all the students in the lab, they contributed proteins and crystals. Thank you for attention. If I can answer any questions you might have.
Okay, please, any questions from audience? Okay, may I ask you a question, John? This is Irina Koshlova. For the data collection strategy, maybe I missed something. So all data collection is defined by your algorithm, or you have to choose something manually? What is actually a role of experimentator in the whole procedure? It was not clear how it is done.
Yeah. So far, we experimented a few strategies that we are interested in. For example, this traveling salesman route, that would minimize the total travel. Of course, that's to the interest of the time. And then that's our favorite strategy. But as I mentioned, to prioritize those crystals might be another one. We also experimented that one. The paired light and dark strategy is potentially another strategy that may have a point. So this is the part of work is not finished. Which one is the best in the end is the best to the science. We left an opening in the software, the plugin, for the users to contribute a strategy or sorting, let's say. Yeah, that can be done in the future. Which one would be the best? I think it depends on each project.
Okay, thank you. Any other questions from?
Yes, I have a question. Do you have a picture about how the vapor diffusion takes place growing the crystal in your device?
A good question. Sorry, I not mention this. If you crystallize protein crystal on this kind of device, it's entirely closed. There's no vapor diffusion occurring. This is a batch method. Okay, this is just a batch method. Basically, you you enclose the entire protein precipitant mixture buffer at a fixed condition inside this device, and then close it. When you seal it, it's isolated from the outside world. The condition should not change, except this device is there's some leak into it. It should not dry out in a reasonably long period of time, say weeks and months. It should not have any condition change. This is called the batch method in protein crystallization. All of our crystal that so far grow is under this condition. All of them are derived from batch method. But of course, in the future, we are actually experimenting different design of the device to retain the advantage of the design, but to introduce either vapor diffusion or hanging drop. These are different features into that. I hope I answered the question.
Yes, thank you.
All right.
Any other questions? Okay, so I have another question for you, quite technical. You know, you are using Z-cut quartz in a certain orientation, I understand, and how difficult to get this crystal in this orientation, and how about radiation damage? Can these cells be reused, or this is one-time deal?
Okay. The Z-cut means normal to the surface is the trigonal, is the threefold axis of that quartz crystal. They call it in this semiconductor industry, this is called the Z direction, the Z-cut. It's commercially available, so that's not a problem. Whether this is disposable depends on how rich you are. Ideally, of course, it will be disposable. The student will be the happiest, so they don't have to take care of the aftermath. But we are not that rich yet, so we encourage them to recycle at least some of them. And successfully, they're doing a really good job, can recycle it. Not 100% though, a very decent percentage of them can be recycled. Because this material nothing is going to do to that material. It's literally just like a glass, except it's made very thin, it may be fragile. Yeah.
Thank you for your answer. Please, other questions to our speaker? Okay, it looks like there are no more questions. I would like to thank you, Zhong, for a wonderful talk. and I hope to see more of your devices at our Beamline available to other users to test. Thank you so much.
Thank you for the opportunity. I enjoyed it. Thanks. Bye.
Also, I would like to remind you that we are searching other speakers to talk about the research at our seminars, and I would like to ask you to contact us if you would like to present your research. Thank you so much, and it was my pleasure to see all of you at our seminar. See you next time, which will be in November. Have a good day now.